Good afternoon, students. Yesterday I told you about two models of homologous recombination. That is number one, the Holliday model, and number two, the double strand break repair model. So let us discuss about Holliday model. What are the general methods of types of recombination? Number one, homologous recombination. Number two, non-homologous or illegitimate recombination. Number three, site-specific recombination. And number four, replicative recombination. Okay. Now, so in detail we are going to study homologous recombination. How does it take place? By Holliday model or by the double strand break repair model, we can explain it. Okay. Let us discuss about Holliday model of recombination. There is a pairing or alignment of two homologous duplexes of the DNA molecule. The method is single strand invasion, in which out of the two strands of the duplex, only one strand is nicked and only one strand invades or one strand moves. Okay, so endonuclease nick at corresponding region of the same strand of homologous chromosome. Okay, so endonuclease nicks at corresponding region of the same strands of homologous chromosome, and it cleaves only or nicks or cut only one of the two strands of the two duplexes. The ends that are generated by nick or cut invade the other duplex. Okay, so if there is, say for example, duplex A and duplex B, out of the two strands of the duplex A, one is cut, and out of the two strands of the duplex B, one is cut, and the cut ends of the DNA migrate to the other duplex. Okay, when they migrate to the other duplexes, okay, a joint molecule is formed, and the nick or cut is sealed by the enzyme ligase, and a joint molecule is formed which is called as Holliday intermediate or Chi structure, and hence the name of the model of recombination is Holliday model. Then there is branch migration, expands, and heteroduplex regions are formed. So let us study the Holliday model with the help of diagrams. Now, if you look at the diagram, you can see there are two duplexes. There is duplex 1 and duplex 2 shown by different colors, right? The duplex A having gene ABC and of course complementary genes A dash, B dash, C dash running in 5' to 3' and 3' to 5' direction. So there are two duplexes, one shown in red color, another shown in blue color. Okay. Now out of the two strands of the duplexes, one of the strand of both the duplex is nicked. You can see here the lower strand here. One of the strands of the two duplexes is nicked by endonuclease. Okay, that is called as single stranded break or single stranded nicking. Okay, so endonuclease enzyme cut the two strands. Out of the two, only one strand. Okay, both of them are homoduplexes as shown here, right? So there is a cut or a nick or single stranded break is created by endonuclease enzyme. Out of the two strands in each duplex, only one strand is cut. Okay. Now the next. When the one strand is cut and the two, as I told you in the beginning, the two, 1 minute. Now the two cut ends migrate towards each other. Okay. The two cut ends can migrate. As you can see in the diagram, they are migrating towards each other, and a joint molecule is formed. Ligase will seal the gap and we get a joint molecule. This joint molecule which is formed is called as Chi structure or Holliday joint. This joint molecule is called as Holliday joint or Chi structure. What happens in the joint molecule? There was a cut here, right? The strands have migrated towards each other, right? The strands have migrated here and one of the strand has come here. So the two strands have exchanged themselves and a cross or you can say X-like molecule is formed and the two branches migrate, right? Now the model, okay, or the Chi model or the Holliday model, you can say isomerizes itself. The Chi model or Holliday model isomerizes. So you can see a X type of structure here which is called as Holliday junction or Holliday intermediate after the name of the scientist Holliday. Is that clear? Now, so you can see here the two homoduplexes are combined in the form of an X. Right? Now the two homoduplexes are going to separate. The two duplex DNA are going to separate. So this Holliday model is either cut in a vertical manner or in a horizontal manner. So it will be cut in east-west direction or it will be cut in north-south direction. So there it will be cut like this. It will be either cut like this or it will cut like this, and you will get two heteroduplexes. How it happens? So let us study with the help of a diagram. Okay. Now resolution of this joint molecule. So we got a joint molecule called as Holliday molecule. It can occur in two ways. The Holliday junction can be nicked in the same strand that were initially nicked. So that is called as horizontal resolution, and it does not result in recombination of flanked marker. So there is no recombination of flanked markers if the Holliday junction is nicked in a horizontal resolution. It has to resolve. Okay. So both the duplexes are now combined and formed an intermediate. So the intermediate has to break and resolve. How will it break? Either in a horizontal manner or in vertical manner. Okay, so if there is horizontal resolution, then the flanking markers do not undergo recombination. But if the Holliday junction is nicked, the Holliday junction that were not nicked earlier results in vertical resolution. And this vertical resolution results in recombination of flanking markers. Okay, let us study this diagram. We can see from this diagram vertical and horizontal resolution. Okay, this one, if it occurs in this manner, it is vertical, and if it occurs in this manner, it is horizontal. Okay, so this is a Holliday intermediate, and now it is going to break vertically as well as horizontally. What happens when there is horizontal breaking? You can see here the original strand remains as it is, AB, where it was nicked earlier, and the B strand remains as it is. I mean, of the other duplex, one of the strand remains and only one strand of the two have changed. So a region of heteroduplex is left, but the flanking markers are not recombined. Okay, the flanking markers are not recombined in case if horizontal resolution takes place. What happens if vertical resolution takes place? If vertical resolution takes place, this leaves a region of heteroduplex and the flanking markers are recombined. You can see this is a heteroduplex recombined, and this is a heteroduplex which has been recombined. Okay, so in case of Holliday model, there is single strand invasion, there is single strand break, which results in an intermediate formation of an intermediate you can say called as Holliday junction or Holliday intermediate, which undergoes resolution either in a vertical manner or in horizontal manner and results in the formation of heteroduplexes. So if it breaks vertically, it will result in heteroduplex having recombined flanking region. If it breaks horizontally, it will have flanking markers that are not recombined. So this is how we can explain the recombination of homologous by homologous method. So again you can see north-south, that means horizontal resolution. So okay, that is vertical resolution. So you can see how vertical resolution has resulted in the form of splice recombinant or recombinant of flanking regions, right, where both the duplexes are now recombinant ones. If it occurs in a horizontal manner or in east-west manner breaking or resolution, then it is patch recombinant, wherein the flanking regions are not recombined. Okay, so this is how Holliday junction cleaves or resolves either in vertical or horizontal manner, leaving patch recombination or splice recombination. Now the other method. Okay, the other method is called as double stranded break model or double stranded break repair. How it takes place. This model provides a better explanation for recombinant events, especially in yeast. What happens here? There is a double stranded break. Okay, the endonuclease enzyme cleaves both the strand of one of the duplex. So there is a double stranded break, means both the strands are broken, and then recombination takes place. One DNA molecule is preferentially used as a donor of genetic information. Okay, there is one duplex wherein break occurs in both the strands. There is another duplex which serves as a donor. The gap substrates can initiate recombination and repair process is required. So now you can see a double stranded break repair model. Right. Look at the first point, first diagram. Here you can see two duplexes. Out of the two duplexes, double stranded break has occurred, or both the strands of one of the duplex are broken. So there is a double stranded break and gaps are created or DNA is removed. Okay, you can see gaps are created in the second, you can see diagram. Double stranded break has taken place and double stranded break resectioning also has taken place. There is loss of nucleotides, loss of small segment of DNA. And if this occurs, this has to be repaired in the DNA. And then how recombination takes place and repair takes place can be explained. Now, look at the third one. There is another duplex DNA which is going to serve as a donor DNA. So, the donor DNA invades towards the recipient DNA, and then the recipient DNA, one of the strand also invades towards the donor. Okay, so you can see in the first there is a nick in two strands. In the second one, some of the DNAs are lost, and then strand invasion takes place. There is strand invasion and repair synthesis starts. Okay, strand invades towards other and repair synthesis starts. Second end capture, synthesis and ligation takes place. You can see the donor DNA, which is shown in red or maroon color, okay, has invaded towards the recipient strand, or where there was double stranded break, and now new strands are getting synthesized. Okay, so against this red color strand, the new strands are getting synthesized, and the donor DNA is going to give the DNA to the recipient one where there was cut, and now resolution takes place. After resolution, you can see about crossing over. No crossing over has taken place, but repair has taken place. So, against, see, so this is how it has invaded, and against this strand, a new strand has been synthesized by DNA polymerase enzyme. New strand has been synthesized by DNA polymerase enzyme. So, there is no crossing over actually, and repair has taken place. But it does not result in recombination, right? And this, the second is showing there is double stranded break repair with crossing over. So crossing over has taken place, exchange of segments have taken place, and the molecule has been repaired as shown in the second part. Okay, so if there occurs break in both the strands and the donor DNA invades, and one of the strand of the recipient invades towards the donor, and there is repair mechanism either by crossing over or without crossing over. Okay, if it occurs without crossing over, then flanking region may not be recombined, but if it occurs with crossing over, flanking region will be recombined. This is how double stranded break in a DNA can be repaired, and by the process of homologous recombination. Okay, so that is all about different types, general types by which recombination can take place.